Muscle fibers. The star of our journey is the muscle fiber itself. These fibers range in diameter from 10 to 100 micrometers. That's a tiny fraction of a millimeter. Some muscle fibers can be as long as 30 centimeters, 12 inches. That's about the length of a school ruler. These fibers are formed during embryonic development when hundreds of smaller cells called myoblasts fuse together. Each muscle fiber has many nuclei because of this fusion. Once they come together, they don't divide anymore, which means the number of muscle fibers you are born with stays the same for life. Sarcoma, transverse tubules, and sarcoplasm. Under the microscope, you would notice that the multiple nuclei just beneath the sarcoma. The sarcoma is like the outer membrane or skin of the muscle fiber. Tiny tunnels called transverse tubules dive deep from the surface into the muscle fiber. These tubules are filled with interstitial fluid and help quickly spread muscle action potentials or electrical signals throughout the fiber. This ensures that every part of the muscle fiber gets activated at the same time. Inside the sarcoma, you find the sarcoplasm, which is like the cytoplasm of the muscle fiber. It's packed with glycogen, the storage form of glucose. This glycogen is crucial because it can be broken down to produce ATP, the energy currency of the cell. Additionally, the sarcoplasm contains myoglobin, a red-colored protein that binds oxygen. Myoglobin releases oxygen when it's needed for ATP production in the mitochondria, the powerhouse of the cell, which is strategically placed close to the muscle's contractile proteins. Myofibrils and sarcoplasmic reticulum. As we zoom in further, we see the sarcoplasm stuffed with tiny threads called myofibrils. These are the contractile organelles of the muscle, about 2 micrometers in diameter, extending the entire length of the muscle fiber. They give the muscle its striated, striped appearance. Surrounding these myofibrils is the sarcoplasmic reticulum SR, a network of fluid-filled sacs similar to the smooth endoplasmic reticulum in other cells. The SR stores calcium ions, Ca, which are essential for muscle contraction. The ends of the SR, known as terminal cistons, butt up against the T-tubules, forming structures called triads. When the muscle needs to contract, the SR releases calcium ions, triggering the contraction process. Filaments and the sarcoma. Inside each myofibril are even smaller structures called filaments or myofilaments. These come in 2 types: thin filaments composed mostly of actin and thick filaments composed mostly of myosin. These filaments are organized into repeating units called sarcomeres, the basic functional unit of muscle fibers. The sarcomeres arrangement creates a variety of bands and zones that give the muscle its striated look. The darker band contains the entire length of the thick filaments, while the lighter band contains only thin filaments. The Z discs mark the boundaries of each sarcoma, and the H zone in the center of the A band contains only thick filaments. The M line in the middle of the sarcoma holds the thick filaments together. Summary. So, to summarize our adventure, skeletal muscle fibers are complex structures formed from many cells that fuse together during development. The sarcoma and T-tubules ensure rapid communication within the fiber, while the sarcoplasm provides the necessary nutrients and oxygen. Myofibrils and the sarcoplasmic reticulum work together to produce muscle contractions, and the filaments within sarcomeres create the muscle's striated appearance.